So hello everyone and welcome to our webinar. My name is Kirsi Kinnunen and I'm a principal biomarker scientist at IXICO, and I will be the moderator for today's session. And as you probably all know, Alzheimer's disease has traditionally been viewed through the lens of amyloid plaques and tau tangles, but as the research has evolved and keeps evolving, so does our understanding of the complexity behind this disease. So increasingly, we are seeing how comorbidities like neuroinflammation and cerebrovascular pathology can play a role in both the risk and progression of Alzheimer's disease. And radiology is really at the forefront of uncovering these connections. Today, we are joined by two exceptional speakers who bring deep expertise and practical insight to today's conversation. First, we'll hear from Professor Frederik Barkhof, who's a leading neuroradiologist whose work has helped shape really how we use imaging to understand many neurodegenerative diseases. And Frederik will walk us through the evolving role of imaging in Alzheimer's trials with a focus on disease subtypes, AD comorbidities and co-pathologies, and imaging biomarkers. Following Frederik, we'll be joined by Alex Henderson, who's a radiology manager at IXICO and who will share perspectives on the operational realities of deploying advanced imaging strategies in clinical trials. And this will include protocol standardization, infrastructure needs, and cross-disciplinary collaboration. We'll also have some time for Q&A at the end, so please feel free to submit your questions throughout the session. We'll look at them at the end. And with that, let's begin. So Frederik, over to you.
Thanks, Kirsi. I hope you can hear me well.
Yeah, we can hear you loud and clear, and we can also see your screen slides.
Yeah, all right, lovely. Well, thanks for inviting me, and thanks for the kind introduction, Kirsi, and welcome everybody to this webinar wherever you are. I will take the next half hour or so to talk about what you can see on the screen. So as Kirsi mentioned, what can we do with imaging? There's obviously some more bread and butter things that we can do. Think of area monitoring, etc., but also things that are only just emerging, and I will focus on that a little bit more. So comorbidity, novel targets, and what does imaging have to offer you now and perhaps in the foreseeable future. Here are my disclosures. I were, the consultancy work for IXICO. I'm also a central reader in many studies, but also work with lots of other pharma companies, partly in the Alzheimer's space and partly in the MS space also. And here's the learning objectives as laid out. So comorbidities are more the rule than the exception, as we'll see also in Alzheimer's disease. And I'll go into what sort of comorbidities they might be, other protein depositions, other neurodegenerative disorders, but also neuroinflammation and cerebrovascular pathology, and some of which we can identify with some effort. Obviously, this is also reflected in the trials landscape. Today, people are looking at other targets beyond amyloid and tau. Think of TREM2 for neuroinflammation, drugs that look at blood-brain barrier and cerebrovascular pathology. And then I will focus on the radiologist's role. So how can we identify comorbidities in AD risk and AD progression from an imaging point of view? Also, we'll briefly mention some other markers that are at our disposal and see where we stand relative to, for example, fluid biomarkers. And what targets can we identify to, if we wanna modify these risk factors, what is on the horizon or becoming available. So just to take you one step back outside the trials, here's the patient journey in memory clinic. This is a series of papers that Giovanni Frigioni and some colleagues published recently. Obviously, wave one, if people come to a memory clinic, you want to make sure that they in fact have a cognitive deficit and not something else, for example, psychiatry. And then soon you will start detailing then the cognitive testing, do some routine bloods, but also imaging already. And then there's a series of clinical syndromes which we can describe, and it's good to realize that we always call this memory clinics, but in fact it's much more than memory. There's also much more than Alzheimer's disease, and Alzheimer's disease is not always memory, but this is just the colloquial term that we use. But people can present with visual spatial impairments, they can present with language impairment and still have AD, but also have FTD. So there's a lot of options there, and imaging can already guide some of this early on, or you can make suggestions about additional biomarkers that should be done, either blood or CSF or PET or SPECT. And then second line biomarkers as well, depending on what the first observations reveal. Let's zoom in on Alzheimer's disease, and then from there on, I'm going to make it simple first, and then I'm going to confuse you, I'm afraid. So Alzheimer's disease, most people believe is driven primarily by abnormalities in amyloid, and we know that, for example, from mutation carriers or people with double APOE4, they always get amyloid pathology, and amyloid is actually a normal occurring protein in the brain. But when it starts to aggregate, it starts to interfere with the function of synapses, and then progressively degrades the dendrites, and then forms these extracellular plaques. And we believe that this then secondarily induces the tau pathology in Alzheimer's disease, which we can see on this post-mortem slide. So this is a coronal slide. You see here the hippocampus and the entorhinal cortex. It's an important piece of tissue in the periapical gyrus that's needed to encode new memory information. So if you're in this early stage, according to Braak and Braak's stage, and you're probably starting to have your first complaints, or you may perhaps have mild cognitive impairment. And then subsequently, it spreads, and you get also neuronal damage, and the tissue shrinks, process called atrophy, which is in a way the shadow of the disease. Means that a lot of things have happened already by the time you look at it. And that's what we see on MR. We can see hippocampal atrophy, but obviously, if there's this much damage, a lot has happened before that, and you're quite late to the game. You can be much earlier if you use amyloid PET, where you can see the amyloid in the cortex. This is a normal amyloid scan where you see some off-target binding to myelin. In fact, some people use it as a myelin tracer as well, but you won't see any uptake in the cortex. So as soon as you start seeing uptake in the cortex, so here it's all over the place, but even if one or two regions only are abnormal, you already call it a positive scan. And then at some stage, the amyloid is all over the cortex. There's a variety of tracers that we can use to do that. All of them, and many of them, are fluorinated, which means that they have a good half-lifetime, and you can produce the tracer here and scan a couple of hours later elsewhere. But the difficulty is that they all have a different range in standard uptake value ratio, so it's very difficult to compare the quantitative values. Hence, the centiloid scale has been developed to put everything on a centiloid scale from 0 to 100. Where 100 is the average AD patient, but in fact, some people will have values higher than 100 if they have really severe amyloid pathology. We worked a lot on stress testing the centiloid concept within the AMIPET consortium that I had the pleasure to coordinate, and where IXICO was part in as well. There's it's well validated against neuropathology, and there's a gray zone of about 12 to 13 centiloids so early on, where people are on the fence of becoming amyloid positive, and that I think is the sweet spot for trials in the future, to see if you can people are in this very early trajectory, whether you can prevent them from getting downstream damage and not becoming demented. If we zoom in on this a little bit more, here's another publication from the Amyloid Consortium. Zoom in on this gray zone. You can see that this is, the earliest reliable detectable change is probably from 10 or 12 centiloids onwards. If you're 15 to 17, then you're likely to be an accumulator, and then if you're over 20, you're very likely to start worsening, and people will be able visually to read it as a positive scan. You've probably seen many of these graphs from trials where they used the centiloid scale, and there were some trials that failed to reach a clinical endpoint in a significant manner. And if you look at two of them, gantenerumab, aducanumab, they actually did reduce the amyloid, but not sufficiently to come below this level of 24 centiloids, which is slightly arbitrarily labeled as an amyloid negative scan. And you can see that studies that were significant were able to bring it down below this level. So you probably need to have a sort of rigorous removal of amyloid before you will see a clinical effect. If you now I'm going to confuse you a little bit more, because centiloid, you think always if it's a high value, then it has to be AD with dementia, and it's true. Virtually all AD dementia cases will have a positive scan. Not all MCIs, which is fine, because some will never convert to dementia, so that's all fine. But it's slightly worrisome, of course, that there are also other diseases with positive scans. And for example, in DLB, that's not unsurprising, but also under the microscope, many patients with dementia with Lewy bodies will have a certain amount of amyloid pathology, but you can see even happening in people with cerebrovascular disease, for example. So you can have amyloid pathology in people who don't have AD. The other thing to note is that not all AD patients are the same, and that's a theme that I'll come back to multiple times. Here's work again from the Amyloid Consortium, where we had thousands of scans, and we did machine learning to see whether there were different patterns of amyloid accumulation over time. And indeed there are, and you will see in a minute that also if you do this with atrophy or with tau PET, you will see the same roughly, that there are at least 3 subtypes and possibly even more, with one subtype starting more in the frontal lobe, and that's actually where you often see first visually the abnormalities. But another subtype starts more in the precuneus, which we know is the hotspot for AD. Then there's also a significant group that starts in a more posterior region, and perhaps these are more the people that look like Lewy body dementia or may have visual symptoms rather than the typical memory or perhaps behavioral. So we're trying to link this a little bit more to the phenotype as we speak. Going back from molecular to structural imaging, this is MR and CT. These are people who were scanned on the same day. So this is the same person, this is the same person. Obviously, we love to look at the hippocampus, and we can visually rate that. We can also quantify the volume and normalize that for you. We can look at the rates of atrophy over time. You could use this, for example, to select people. In a memory clinic, we use this all the time. But even in a trial, you could use this by, for example, either excluding people with a completely normal hippocampus or excluding people with a devastated hippocampus. Depends on what your outcome measure is. You can narrow down your population and fine tune it, or at least confirm the diagnosis of AD. But then there are also patients who have Alzheimer's disease, but have a very different pattern. Here's a very young patient who was only 50, came to our memory clinic with a bad MMSE score, had very beautiful normal hippocampi, but look at the atrophy that occurs here in the parietal region. So a very different pattern of atrophy, and this is called posterior cortical atrophy, which is not just a radiological term, it's a clinical term where people have mostly visual spatial disturbances, so they have difficulties navigating around their own house or finding things. Their memory can be relatively preserved, and then so is this clinical presentation with atrophy in the posterior regions of the brain. collectively referred to as posterior cortical atrophy. Can be due to other diseases, for example, DLB, but most of them have Alzheimer's disease. And in this case, also, we did CSF analysis and confirmed there was abnormal amyloid and tau, leading to a diagnosis of AD. So the younger the patients, the more likely you are to see this posterior cortical atrophy pattern. So here I made a summary of these the patterns that we can find. The typical one is the older patient who is ApoE4 positive, has memory problems. They will have hippocampal atrophy, whereas the PCA pattern will be younger patients who are ApoE4 negative, have visual spatial disturbances. But beyond that, there are even more atypical patterns, I would say, where there's a frontal or occipital pattern, or lateralized, left hemispheric, more language type of pattern. And then obviously you run into a differential diagnosis with frontotemporal dementia or Lewy body dementia. So here's a study where we identified in a large group of memory clinic patients with the diagnosis of AD, confirmed by biomarkers. So they all have confirmed amyloid pathology. And we subclassified them by the primary domain affected. So the memory obviously is the most typical one, but then people who have predominantly executive function disorders, predominantly language, predominantly visual spatial, etc. And then we looked at the atrophy patterns, and you can see that the memory one, as expected, shows relatively isolated atrophy of the hippocampus. But people with other domains have much more widespread abnormalities, with the language one being mostly in the left temporal region, as you would see in FTD, for example, and then the visual spatial and the multiple domains have more widespread, including these precuneus and posterior regions that we talked about before. Likewise, we can use tau PET to look at subtypes. These, again, are patients with a specific clinical presentation who had a tau PET with the AV1451 tracer, which is a fluorinated tracer. And in typical amnestic Alzheimer's disease, like for MR, you see that the atrophy or the tau uptake is mostly in the medial temporal and to a lesser extent also in the lateral temporal regions, so the classical patterns. But then people who have more a language variant, and in Alzheimer's you see a logopenic variant of primary progressive aphasia. You see more widespread temporal parietal abnormalities. Here's a PCA pattern, which, as expected, has this very focal abnormality in the posterior cingulate and perhaps even in the occipital lobe. And then there's some other subtypes with bizarre distributions of tau. And these are all patients who fulfill the clinical diagnosis of Alzheimer's disease. So they have abnormal amyloid and they fulfill the clinical criteria. Just to make the point that Alzheimer's disease is not one disease, there are many subtypes of it. I've shown you so far a couple of tracers. The amyloid one is very good to to show amyloid, but we've seen already that people in the normal population, maybe amyloid positive, have no complaint at all. They are at a higher risk to develop a complaint in 10-15 years, but they don't have to have Alzheimer's at all. And conversely, there are people with other diseases who can have some Alzheimer pathology as well. So the most useful finding clinically is to have a negative scan, because then you know that Alzheimer's is definitely off the cards. Whereas with tau PET, that is more typical for AD, and if you find it in the typical regions, that is a very strong positive predictive value. So this is more a rule-in test. Amyloid is more a rule-out, and then FTG is a different type of tracer, because you always have high uptake, high metabolism in the cortex, and depending on where the hypometabolism is, for example here, this is more what you would expect in Alzheimer's disease. If it's frontal temporal, you would expect more frontal or anterior temporal hypometabolism. So this is more for the differential diagnosis. I'm sure you've heard of the ATN staging scheme, which I'll talk through in a minute. Meanwhile, a new set of criteria has been proposed, which relies more on only the amyloid. I'm personally not too big a fan of it, because it sort of overrates the amyloid pathology. Let me first mention this one, the previous one to you. So this is based on amyloid, tau, and neurodegeneration. Whereas in this scenario, if you have just amyloid pathology either on CSF or on your PET scan, it's considered Alzheimer's pathological change. If you then also have tau pathology, it's called Alzheimer's disease. If you then also have dementia, it's called Alzheimer's dementia. But obviously there are people with other combinations which make you wonder what's going on there, and I'll go into that in a second. As I said, there's been a recent proposal to change this and call anybody with amyloid in their brain Alzheimer's disease, which doesn't go well with a lot of clinicians, because if people are not demented, why would you call them diseased? They have a disease process perhaps, but they don't have an illness. So there's a fierce debate around this evolving nomenclature. Just briefly to say that all of this can be done with imaging, but also with biomarkers, either in CSF or progressively also in blood. For example, the tau markers in blood, p-tau217, for example, has pretty good performance. The amyloid blood tests are lagging behind a bit, but are coming there. And obviously we can measure some other things in blood as well. So coming back to the ATN staging, what you would expect in an Alzheimer patient is to have an abnormal amyloid scan with the amyloid in the cortex, then a positive tau scan with either abnormality restricted to the medial temporal lobe, with a lateral temporal, or perhaps already to the parietal, and then atrophy. So you have ATN positive. That's all fine. If you find somebody very early, and perhaps they may even be asymptomatic at this stage, you can see an abnormal amyloid, because we cannot see the white matter and the cortex separate, so it's a positive scan. But then the tau is negative, the MRI is negative, so this is just preclinical Alzheimer's pathology. But then there are really confusing situations where people are amyloid positive, have a negative tau scan, but still have massive atrophy, including the hippocampi. And this is called suspected non-Alzheimer pathology, a very descriptive term. And here's one such case from Cliff Jack's lab that had a negative tau scan, was amyloid positive, had massive atrophy. And this was an older patient with progressive amnestic dementia and had been diagnosed as Alzheimer's disease. But in the end, it turned out that it was all due to hippocampal sclerosis, or TDP-43 pathology. For which now there is another term, limbic predominant age-related TDP-4 encephalopathy, or late. And this is extremely confusing clinically, because the patient has dementia, mostly amnestic, and his hippocampal atrophy. So you would say it's a slam dunk, but in fact it's all driven by another type of pathology. And this is a very confusing entity. It worries me a lot, because we have no good measure for TDP-43, neither in blood nor in PET imaging. Also, other diseases that can affect your hippocampus. For example, here you see a patient with very severe hippocampal atrophy, but note that it's quite asymmetric, especially if you go further posteriorly. And if you see very asymmetric hippocampal atrophy, you immediately have to think of FTD. And in fact, this was a patient with semantic dementia, is one of the variants of FTD. And the only thing that you might if it's so end stage, it has to be FTD. There's no doubt about it. If it's slightly less marked, you could think of the logopenic variant of Alzheimer's disease, which can be left temporal dominant and have language problems as well. But again, you need biomarker data to be able to tell them apart in some cases. There are also other subtypes of FTD. I'm not going to go into that too much. Behavioral, so the semantic dementia we already mentioned, non-fluent aphasia, and the right-sided FTD, which have predictable patterns of atrophy and predictable patterns of hypometabolism on FDG PET. The difficulty with FTD is that we don't have very good molecular biomarkers because these are sort of indirect effects. You see atrophies, your hypometabolism, but to show the causative proteins and to make a positive confirmatory test in FTD is very difficult. only 10 to 20% of people will have a genetic mutations. The other are unknown variants. And it's often a diagnosis by ruling out. But I do think, you know, even in AD trials, we not infrequently see patients that I'm 100% sure that they have FTD and not AD. So you have to be careful with that. So here's a list of the common disorders if you have a patient with dementia. The whole list is massive. I don't have time to go into that. But you know, we'll talk a lot about AD. I already mentioned FTD. And then there's patients who have cognitive problems and dementia with Parkinsonian features, notably dementia with Lewy bodies. There's multisystem atrophy, and there's PSP. And all of them have Lewy body pathology and can be diagnosed with a DAT scan. And then there's vascular dementia, which pure vascular dementia is perhaps rare, but there's a lot of subclinical cerebral small vessel disease, which probably adds to the severity of the presentation. And I will go into the interplay between that and AD in a minute. If you look at the prevalence of the various disorders, so about half of the patients have Alzheimer's disease, and then, well, this is just one study. You can find endless studies, and you know the percentages vary depending on the definitions and the population that they studied. And here they claim that, for example, 20% is cerebrovascular disease, probably overrated. And what is also strange, you know, there should be an overlap between the two. In fact, if you zoom in on the right side on the AD part, is less than 50% have only AD at pathology, at autopsy. And then a quarter has AD and vascular dementia. Another 50% has AD and DLB. Then you have 7% have AD and DLB and VAD. And then, well, it goes on and on and on and on what combinations are possible in a sample of people that fulfill criteria for Alzheimer's disease. And here's another post-mortem study where they looked at the number of neurodegenerative pathologies found, which can go up to 7. And you can see even in unimpaired, cognitive unimpaired people at autopsy, you can find all sorts of changes. Some diseases are relatively clean, like motor neuron disease, FTD, perhaps MSA. But if you look at clinical AD, it's the exception to have only one pathology. And you see that the majority has multiple other pathologies going up to 7, which is really scary. And here's another cartoon showing that you know, certain proteins can produce multiple diseases, and that multiple pathologies can occur also across the diagnostic spectrum. So if you're in the Parkinsonian part of the spectrum, then it's very useful to do a DAT scan, and the DAT scan in healthy controls will nicely show you this comma-shaped appearance with the caudate and the putamen, and obviously you will also have normal hippocampi. If you have DLB, then the dopamine is gone. You probably have relatively good hippocampi, and if you have AD, you have hippocampal atrophy, but nicely preserved the dopamine uptake in this striatum. So this is extremely helpful, and the DAT scan is also useful in MSA and PSP as well. So this is a very useful molecular piece of information. I've talked mostly about imaging because that's my particular cup of tea, of course, but it's fair to say that a lot of things can be measured in CSF and progressively also in serum. Amyloid, as I mentioned, amyloid is a naturally occurring protein. So the abnormal finding is that it's gone down rather than up. Whereas all the others, normally you don't have them. So they're normally absent, and then they go up if they're at normal. So tau, we have a series of good candidates there. For alpha-synuclein, it's coming a little bit, especially in CSF. We can now use this seed amplification assay. Not yet very well in blood. And then there's a series of other interesting markers for neurodegeneration at large and inflammation, especially GFAP is getting progressive traction. Also here, good to mention that for missing markers is for TDP still. And then you've probably seen some cartoon like this, where the sequence of events is portrayed with amyloid happening early, probably in the wet biomarkers a bit early than the PET, so they plateau relatively early, and then the PET continues because it shows more the spread of the amyloid through the brain. Likewise, the wet biomarkers for tau are earlier than tau PET and then plateau, whereas the tau PET keeps tracking the spread of the pathology, and the same is for atrophy and then cognition. So this is the window for early symptomatic AD trial, where you would probably have some degree of tau PET abnormality already. So yeah, and a lot of these things that I mentioned affect also the heterogeneity within a trial, and obviously we all try to reduce that at the danger of restricting the final label. But on the other hand, you want to have a well-defined population to target a drug. If it's too diffuse, then you're probably mixing in too much other pathology, too much heterogeneity. So classical ones are, in AD, the number of microbleeds, or excluding massive cerebrovascular disease, or perhaps narrow down on amount of atrophy or an amount of tau to sort of better define the population. But even then, there's a lot of natural biological heterogeneity. So there could be different patterns of atrophy, so more the subtypes. There could be different stage of the atrophy within that subtypes, and I think we could make better use of that because these people obviously will have different risk of progression and may require different outcomes even, and so short of stratify for it, you could use that as a covariate in the analysis. And then there is co-pathology that will affect the clinical outcomes. Other neurodegenerative pathology, think of Lewy body admixture, think of people with trauma, and then vascular pathology. And then more subtle things are emerging, like blood-brain barrier disruption and neuroinflammation with uncertain roles, but probably very early events already. So to briefly mention the cerebral small vessel disease, which you can measure nicely on a flare with a simple scale from punctate to early confluent to confluent. And if you follow people who are living at home and just follow them over a couple of years and see whether they're still ADL independent, you can see having a high Frederik score is very unfavorable. And many of these people lose independency or even die. Not necessarily because of stroke or dementia, because they also have cardiovascular risk, so they could have renal impairment, they can have cardiac, myocardial infarction, etc. But even if we look in the normal appearing brain tissue, so now we're looking outside these white matter lesions. If the histogram, here look at histogram analysis of ADC values from diffusion tensor imaging. If you're in the lowest percentile, so you have more diffusely abnormal brain tissue, you're much more likely to decline than if you're in the highest quartile of this. And this is independent of your Frederik score, of lacunes, and other stuff. So there's probably a lot more widespread pathology going on in the normal appearing brain tissue than we think if we just look at the scans. We've recently looked at that a bit more in detail with DTI and looked in specific tracks. It's too complex to go into the details, but if you look at people with amyloid or amyloid plus tau, you can find abnormalities in certain tracks. If they have cerebral small vessel disease, you can see a very different pattern. And in fact, it's more the anterior tracks that are affected in cerebral small vessel disease, whereas the tracks for AD are more in the posterior regions, also because that's where we see the early tau pathology, of course. So this white matter vulnerability is not random. And what drives this? What drives all these normal appearing white matter changes? Well, the blood-brain barrier is undergoing alterations with aging, in normal aging and in AD. Various things happen and make this more leaky. And cells can go, so either antigens and other things can go into the CNS, but also cells can migrate into the CNS that shouldn't be there. And we can now, there are various techniques coming to the fore that will allow you to measure the degree of blood-brain barrier disruption. We've worked mostly with an ASL-based paradigm. So here you see the ASL maps, which usually used to measure perfusion, and the perfusion declines as we grow older. But if you do this with multiple echo times, you can also see that the diffusion of the tracer across the blood-brain barrier, you can map that. And here also you can see that that goes down with age. There's slightly stronger age effects in CBF, and slightly stronger gender effects, but also here you see the time of exchange across the blood-brain barrier goes down as well. We can also try to characterize in a molecular manner what happens beyond amyloid and tau that we already discussed. For example, you can look at neuroinflammation with TSPO, which is thought to be mostly on activated microglia. You can look at neuronal density or synaptic density with tracers. And this brings us to the role of inflammation in AD, because we see activated microglia, and there's quite a bit of a debate whether this is beneficial or deleterious. Probably a bit of both. On the one hand, it may be a result from the damage occurring, but to a certain extent it may also in itself drive it. And then you have people with mutations who have more severe AD pathology. People are also developing, for example, antibodies against TREM antibodies. So this is an interesting pathway, but it's not completely well understood how early and how causative these microglia changes are. Likewise, we can look at astrocytes, which with Deprenil, we can image the amount of activated astrocytes. And again, they are increased in AD. And the group of Agneta Nordberg has looked at this in various stages of the disease. And they see positivity relatively early already, almost before the amyloid, or perhaps when the amyloid starts building up. And then subsides, and then in a later stage you again see an increase. And this is more around the time where also GFAP becomes abnormal, which is a wet biomarker for activated astroglia. So there's much to be learned still about the role of inflammation, perhaps initially protective and then later stage not. I think that's still up for grabs. Zooming out for a moment now to vascular pathology and thinking from a structural perspective. Some people have pure vascular dementia with extensive white matter lesions, lacunes, but then very nice hippocampi. But it's in fact quite rare. Which is much more common is to see this extensive white matter lesions, some cortical infarcts, but then also very severely affected hippocampi. So this is a mixture of vascular and AD pathology, and as I mentioned, it's not unlikely that this patient at postmortem would have even more types of pathology. And this is a really interesting topic, the interplay between vascular and AD. I could do the whole lecture only about this. Obviously, if you have two pathologies, it's not good for your brain. And in fact, only people who have multiple pathologies are demented during life in large population studies. But it's also true that if you have atherosclerosis, you accelerate the formation of amyloid. Whereas conversely, if you have amyloid pathology, it will also be amyloid pathology in your vessels, which will then compromise the perfusion and then lead to vascular pathology. So it's a sort of vicious circle where so these are not two completely independent diseases that lead to cognitive decline, but in fact, one pathology can lead to both and conversely. So they can, in fact, it's a circle between the two. And one of the links here is microbleeds that you can see in both in hypertension, but then in a different pattern in amyloid pathology, CAA and AD. So lots of information. I'll try to summarize it a little bit for you. So structural imaging, I think is important to rule out all sorts of things, to help characterize the severity of damage, perhaps find subtypes, find co-pathology, especially cerebrovascular, but we've seen some others as well. The molecular imaging, of course, is much more specific and complementary to the wet biomarkers. And we can now much more better characterize populations based on amyloid, based on tau, and perhaps some other markers as well. But it's good to realize that some co-pathologies we cannot easily exclude. And then the ATN staging I've talked about, it helps, to have a common language and also sort of biological severity scaling. And we have the centilloids and the centowers for tau are coming, where we have a common language on how to describe our sample, to compare also between trials. And then there's a series of emerging targets. I showed you some examples from blood-brain barrier and tracers for neuroinflammation, but we need to understand better still on what they signify. But for smaller proof of concept studies, these can be used if you have a specific agent that will target the blood-brain barrier or the vessel wall or the neuroinflammation. There are potential readouts for that as well. And with that, I'd like to thank you and stop my presentations, and apologies for overrunning a little bit. One always gets carried away.
Thank you, Frederik, for this very engaging and information-packed presentation. So let's now hear from Alex.
Hiya, thanks Kirsty, thanks for that Frederik as well. Hi everyone, so in this part of the presentation I'll just be focusing on the role of central reading for eligibility and safety reporting in AD clinical trials. So I'll walk through how standardization, workflow discipline and quality oversight all come together to produce eligibility and safety data that's reliable, reproducible and acceptable to regulatory authorities. So like I said, the focus will be central reads, but I will touch on standardization of image acquisition and processing for a bit of context, as they're key aspects that lead into quality central reads. But we do have previous content on this in one of our earlier webinars, which I would encourage you to check out if you're less familiar. So yep, the first slide, start with a very quick summary of what central reading is and why we use it. So put very simply, central reading is a process designed to ensure consistency and enhance the objectivity and imaging assessments across all sites of the study. So instead of having local radiologists interpret scans independently, all the imaging data are transferred to a central reading facility like ICSICO, where trained experts such as Frederik and his team will work under harmonized conditions to perform the reads according to standardized criteria. This setup will just eliminate much of the variability that can happen when we have different sites, scanners, and readers interpreting images in a different way. It's also something regulators increasingly expect in AD. So both the FDA and the EMA look for standardized and auditable central reads when reviewing Alzheimer's trials, because they know the consistent methods will translate into more credible results. So basically, in a nutshell, central reading ensures that every image is assessed fairly and objectively, and it also enables fair comparisons across participants, sites, and time points. And if we go to the next slide, please, Jane, you'll see why that's so important for AD trials. So standardization and why it's so critical. So Alzheimer's disease trials are typically large, they're multi-site and multinational or even global. So this means that there's dozens or even hundreds of scanners, operators, and imaging protocols involved. And each of these factors introduces a potential source of variability. On top of this, then, we have reader subjectivity, and this adds another layer of variability. It's very possible that two highly experienced readers may interpret the same scan differently, particularly in borderline cases, for example, in PET reads for amyloid positivity. So these most common sources of variability can be categorized as we see them in the diagram here at the bottom, and then procedures introduced for each of these to harmonize them. So acquisition through site qualification with defined imaging protocols and the use of phantom scans. And in data handling, this will be done using structured QC workflows and secure storage of the data and transfer. And then in reader training, so through initial qualification during the onboarding of readers, and then periodic requalification, which can help prevent drift over time, especially in long studies. PET readers will complete tracer manufacturer training where it's available. And then all readers will also undergo project-specific training. And this includes the clinical protocol, the study-specific read forms, and also the reader manual, which I'll give a little bit more detail on later. So all these elements together ensure that the trial outcomes that we see, that they reflect genuine change rather than any inconsistencies in the methodologies. So next slide, please, Jane. So here we have the central read workflow and how the process operates in practice. So the data is acquired by the site, uploaded to the central storage system, and it undergoes QC. So the QC is essential to make sure the data meets the requirements for read. In AD, obviously with an older population, motion artifacts are very common, and particularly as the study progresses. So we want to make sure that the data that we send for read is the best quality, and if possible, we can get rescans, and then provide the best quality to the data or to the readers. The readers then interpret the images according to the study's criteria. The criteria are defined at the beginning of a study, generally in consultation with the sponsor, imaging vendor, and ideally a consulting neuroradiology expert within the field. It's a really important stage. This is where we make sure the needs of the study are met by thoroughly reviewing the clinical protocol and covering any MRI-related inclusion and exclusion criteria in the eligibility read form. So for MRI, this will typically include assessments such as the presence of microhemorrhages, the number and the location, and Fazika scoring for deep white matter lesions. And then, so the safety assessments in AD will typically focus on ARIA, ARIA-H and ARIA-E, and also include grading scales such as the SSAE, so the 3-point or 5-point severity scale for ARIA-E. And this will be agreed with the sponsor when developing the forms. While it can sound potentially trivial, it is vital that we include all the relevant assessments in the read form for a number of reasons. So we'll include an assessment that will capture anything completely unexpected. But for the findings that we anticipate, such as hemorrhages and edema, we want detailed assessments on the form, and primarily so that we can provide consistent and comparable data. The aim is to minimize the need for our readers to provide free text, and this also increases efficiency for them in completing the read, and then in turn supports us in meeting tight turnaround times. Next slide. So another aspect of the read workflow that will be defined at the beginning of the study is the read paradigm. So there's a few common models. Firstly, there's the single reader model for straightforward endpoints and can be used for ARIA monitoring on MRI. Then there's the consensus read model, in which multiple readers will discuss and agree a final interpretation collectively, which can be useful in both MRI and PET reporting for difficult or borderline cases. Lastly, there's the dual reader model with adjudication. This is where two independent reads are compared and a third independent reader is introduced when there's discordance between the two reads. This reader either resolves the discrepancies or provides an additional read for a majority. We see this model used in amyloid PET, where we have binary outcomes of amyloid positive and negative classifications, and when they're used as primary endpoints. And it's also expected by regulators, especially for registration grade studies. So the chosen paradigm or model will depend on the endpoint. So is it safety? Is it efficacy? It'll also depend on the level of subjectivity involved in the read, and then ultimately as well, the regulatory expectations for the trial. In any model, it's imperative that all reads are conducted within a secure and auditable system that's also compliant with regulatory requirements that were noted on the last slide. And this ensures the data integrity. For adjudication reads, we need to make sure there's a purpose-built system that will ensure the independent reads are blinded to each of the readers, since the same data will be reviewed and we need to prevent bias. And then the final consideration for a paradigm, for which paradigm to use, is efficiency. So most central reads will aim for a turnaround time of just 2 to 5 days from the data upload to the report and the results. So we need to make sure this can be met with the most appropriate model, which supports timely decision making and to make sure things are kept on track in the study. So the next slide. And another aspect that we want to consider in AD trials is hybrid reading, where both qualitative and quantitative assessments are combined to provide the final outcome. So the visual or qualitative reads rely on the expert judgment. For example, a reader assessing an amyloid PET image to determine whether the uptake is consistent with pathology or the MRI atrophy patterns are consistent with AD progression. So the visual reads can be complemented by quantitative analysis, which provides objective metrics such as SUVR and centiloid for PET and regional brain volume or cortical thickness for MRI. These measurements will help us track subtle changes over time that might not be obvious visually, and they can assist the reader in borderline cases. So as we saw Frederik presented earlier, one of the slides, he spoke about the centiloid and that evolving amyloid range that includes a range that predicts accumulation prior to it being visible on a PET scan. So the centiloid value in these specific instances can be really, really useful for the reader. So, and as such, increasingly, the hybrid approaches are being used for amyloid PET, where quantitation assists the visual reader. However, the final interpretation always rests with the trained expert, as the quantitation measurement may not always be accurate. There may be overspill of uptake into the white matter or suboptimal regional segmentations that obscure the SUVR or centiloid. So it's important for this reason that, the quantitation is used as an adjunct to the visual reads. Overall, the hybrid model combines the efficiency of automation with the nuance of human clinical judgment. So it helps us be faster, more objective in our reads without losing any clinical interpretability. And next slide, please. Here we have quality assurance and oversight. So we put all the features discussed in place, and we need to then make sure they're maintained throughout the life cycle of a study. And this is done through continuous quality assurance and oversight. To maintain consistency across a pool of readers, inter-reader variability is monitored at regular intervals, and this will typically be annually. This can help identify if recalibration is necessary to make sure we're still in alignment with the study criteria. We'll also closely monitor turnaround times, so TATs, to ensure our workflows are efficient and that our bandwidth is sufficient so that we adhere to the project timelines. And then we also have comprehensive documentation. It's maintained at every stage of the workflow. For central reading, we need thorough training guides to our systems and image viewers. We need general operating procedures ranging from the onboarding and selection of our readers to their assessment and monitoring. And then we have the project-specific material that I mentioned earlier, clinical protocol, and the reader manual. And the reader manual summarizes all the relevant study information from the protocol and details all the reading criteria for that study, from expected findings and grading where applicable, and also how to complete the read forms in the system and what logic the readers can expect to be implemented on these. We also detail the contested read workflow for each study in the reader manual. This is used in cases where two readers disagree or the result is ambiguous, which leads me into my last slide. You can just move on one, please, Jane. Challenges and lessons learned. So despite the rigorous practices and associated documentation we put in place, challenges always crop up. The first and most prevalent challenge is protocol deviations due to the diverse networks of global imaging sites that I mentioned at the start. It can even occur if a scanner just undergoes a software upgrade and we're not notified. So our procedures encourage sites to proactively inform us of upcoming changes or deviations, and then we can then manage this, document it, and get guidance from our readers on how best to proceed. So, for example, if a follow-up subject visit must be scanned on a different scanner, we'll check with them if this is likely be comparable to the previous visit to assess any change. Or if nothing can be done, we can inform them of the deviation to make sure they're aware, they can assess the images and don't send them back, and then we remain within our timelines as well. We also have the balancing act of rapid turnaround times with read quality. One way we try to address this is by implementing read form logic in our systems. This adjusts responses on the read forms based on the input from the readers, which both increases efficiency in completing the read form, and it also decreases the risk of any input errors when there's lots of options available for the reader to select from. So lastly, and most broadly, effective communication across CROs, imaging vendors and radiologists, study sites and sponsors is essential, basically all key stakeholders. And this will be from the start of a trial to finish. There's increasing demand for the radiology expertise and consultation during these studies. And this can be to review complex cases where we need to schedule a call. We bring in Frederik, the sponsor, and site where applicable to discuss any findings. It's often needed urgently when there's an SAE. And similarly, readers can assist sponsors with DSMB meetings, from preparation of the safety MRI findings summary and attendance to the meeting to discuss what they've observed and how this might relate to any clinical data that's presented. So I mentioned the last slide, contested reads. It's also key, communication's key here. These will typically need to be reviewed swiftly to make sure we have the right actions taken based on the findings that are identified. Our contested read procedure, which is included in the reader manual, can include a consensus read or an adjudicated read depending on the study, which will be agreed with the sponsor at the beginning. It requires fast action from all parties, and to making sure this is all our processes are documented and contingencies are defined. We can turn these around swiftly and make sure all relevant parties are kept in the loop throughout the process. So that's my final slide. Basically, to wrap up, the well-structured and appropriate central reading process built on standardization and robust workflows and ongoing quality oversight is essential for reliable and credible imaging and reporting in Alzheimer's disease. Now I'll finish there. Thank you very much.
Thank you, Alex, for sharing these insights with us. So we are now nearly out of time, but maybe just a quick question. So this is to Frederik. So how would you say that image quality might affect the ability to detect the type of AD a patient has, and how it could be improved to provide a better, more accurate diagnosis?
Yeah, so it depends on how fine-grained you will be. If you have a proper T1 weighted scan, you could probably look at atrophy patterns relatively well. If you want to portray the amount of vascular co-pathology, you could do that. If you want to be very detailed for example, with vascular, and you want to look at cortical microinfarcts, which I couldn't discuss, then you would need additional scanning. If you want to see more subtle co-pathology, like the normal appearing brain tissue changes or the blood, where you need additional sequences, which can be sensitive to specific disease, but also more sensitive to motion, etc. So it's a bit of a balance, how fine-grained you would like to be.
Yeah. Then there's another very, very describing this time that we're living in kind of question. So do you think advanced AI tools will soon play a major role in differentiating Alzheimer's from other neurodegenerative disorders?
Yeah, well, AI will solve everything, but not in my lifetime. So yeah, it depends on what sort of AI tools are you thinking of. If it's just image analysis, there are interesting things happening, people trying to predict amyloid positivity from structural MR scans, which I find mind-boggling, and I don't truly believe that. But other things are better. It's at the moment slightly better at lower level tasks, quantifying the amyloid load, quantifying the number of microbleeds, etc. For more complex tasks, it also needs more complex input data, which may not be easily achievable.
Can you see a role for AI tools in reads? Alex mentioned something in her presentation.
Yeah, so there are some tools out there. For example, think of simple things like microbleed detection, or cerebrovascular disease segmentation or prediction of Fonseca scores. It's not always perfect, but you could use it as a sort of second reader and say, well, if it's discrepant, please document why it's discrepant and why you would overrule it, or even as a QC step, you could use it. I like that what you presented, Alex. It's a good way to improve the QC, I think.
Yeah. Okay, so that's now concluding this Q&A. And if you have any questions, you can always get in contact with us, either via our website, where there's a contact us form, or Jane will tell you a little bit more. So over to you.
Yeah, I will. Thanks very much there. So if in case you haven't, if you missed any of the webinar beforehand, don't worry, we'll send the recording over to you. Please do follow us on LinkedIn for the latest neuro advances as well. But if you have any questions, if you've sent any to us and we've run out of time, don't worry, we'll get back to you and answer those questions via email. I'd like to thank the panel, and I'd like to thank Kirsty, Frederik and Alex for your time, and also to thank you to the audience as well. Have yourself a fantastic afternoon, evening or morning, no matter where you are and what you're doing. And thank you again for joining this webinar.
Thanks.
Thank you.
Mm-hmm.